Welcome, fellow knowledge seekers, to another deep dive. Today, we're tackling a really big subject. It touches on those fundamental questions, where we come from, how life on Earth actually developed. We've gathered a pretty diverse stack of sources here, scientific journals, research institutes, some thought-provoking articles, and even, interestingly, a fresh look back at Darwin's own writings. So, yeah, get ready to challenge some assumptions.
Yeah, definitely. It's always fascinating how our picture of life's history is, well, constantly being updated, refined. And what's really interesting here, I think, is that a lot of these critiques, these questions, they aren't coming from outside the scientific community. They're coming from within.
Right, from the scientists themselves.
Exactly. As new data pops up from genetics, paleontology, biochemistry, you name it. So we'll be exploring where these challenges crop up, trying to help you connect the dots between these different fields. It's a story still very much being written.
Okay, sounds good. Let's unpack this then. Let's start right at the very beginning, the chemical origin of life. Now, most of us probably remember that classic story, the primordial soup from, high school science class.
Uh-huh.
But our sources are suggesting there are some, well, pretty significant issues with that whole idea. It seems it's much harder to cook up than we thought.
You've definitely hit on a key area of debate. So, problem one. There's really no viable mechanism that anyone's agreed on to actually generate that primordial soup in the first place.
Yeah.
You mentioned the Miller-Urey experiments from '53. They were famous, seemed to show amino acids forming.
Right, the sparks in the flask thing.
Yeah, that one. But crucially, they relied on what's called a reducing atmosphere. Basically, lots of methane, ammonia,
Yeah.
stuff that readily donates electrons. But the thing is, modern geochemists, they largely think the early Earth's atmosphere wasn't like that. It was probably mostly carbon dioxide and nitrogen from volcanoes. That's more of an oxidizing atmosphere. And it just wouldn't support those same chemical reactions needed to make the building blocks.
So the starting conditions were likely wrong.
It seems so. Nick Lane, a biochemist at University College London, he puts it pretty bluntly. He says the primordial soup theory doesn't hold water, and it's past its expiration date.
Okay, so if the atmosphere wasn't right, what else? You mentioned other issues.
Well, yeah, there's an even more fundamental chemical problem, water.
Water, but isn't that essential for life?
It is now, but for originating the complex molecules, water actually tends to break down the long chains, the polymers like proteins and DNA that life needs.
Oh, right, hydrolysis.
Exactly. So trying to form these long chains in a watery soup or even near hydrothermal vents underwater, it's thermodynamically unfavorable. It goes against the chemical grain, you could say. It needs energy input. It doesn't just happen.
Wow, okay. That is a significant hurdle. So, if the soup wasn't really there, and even if it was, making complex molecules in water is really tricky. How on earth did life's, instruction manual, the genetic code, how did that come about? That seems like the next huge question. Unguided chemistry, explaining that seems tough.
It is. That leads us straight into problem two. Unguided chemical processes just can't explain the origin of the genetic code. The main idea floating around is the RNA world hypothesis. The thought is maybe RNA molecules did both jobs, initially stored information like DNA, and acted as catalysts like proteins.
Kind of a precursor molecule.
Sort of, yeah. But there are big challenges. For one, RNA doesn't just spontaneously assemble into functional forms in lab experiments, not without researchers, guiding it very carefully.
So it needs intelligent intervention in the lab.
Well, yeah. As Robert Shapiro, a chemist from NYU, pointed out, the researchers control the experiment in ways that just wouldn't happen on early Earth. It's a bit of a leap to assume that kind of control was available then. And maybe even more critically, there's no real evidence RNA could actually perform all the complex jobs modern cells need proteins for. It's a stretch.
And isn't there just a problem of, the odds? Getting a functional molecule by chance?
Oh, absolutely. The numbers are staggering. For a functional RNA molecule, maybe 200, 300 units long, the odds of that forming randomly are estimated at something like 1 in 10 to the power of 150.
10 to the 150. That's astronomical. Doesn't even cover it.
It really doesn't. I mean, there are only about 10 to the 80 atoms in the whole observable universe. So yeah, the odds are incredibly, incredibly small. Vanishingly small.
Mind-boggling. And then there's that classic chicken and egg thing, right?
Yeah.
With proteins and genetic info.
Exactly. You need complex proteins to process the genetic information stored in DNA or RNA. But the instructions to build those specific proteins are encoded in the DNA or RNA itself.
It's like you need the machine to read the instructions, but the instructions to build the machine are only on the disk you need to read.
That's a great analogy. It's a real bootstrapping problem. How does that system get started? George Whitesides, a top chemist at Harvard, admitted back in 2007 when asked about the origin of life, he basically said, "How? I have no idea."
Wow. Okay, so the key insight here, maybe, is that the very beginning of life, from the chemicals to the code, faces these huge chemical and probability problems. And current unguided models, well, they really struggle, leading top scientists to admit they just don't know. That's a major takeaway. Okay, so from chemistry, let's zoom out. Let's look at the bigger picture of biological history, the fossil record. This is where Darwin himself apparently had some of his gravest objections to his own theory.
He did, yeah. Darwin expected, or at least hoped for, a smooth succession of fossils. Showing gradual changes, lots and lots of intermediate forms. But what the fossil record actually shows, well, often it's the opposite. Which brings us to problem 3. The abrupt appearance of species in the fossil record doesn't really seem to support slow, gradual Darwinian evolution.
The Cambrian Explosion being the most famous example.
Exactly. Around 550 million years ago, suddenly, almost all the major animal body plans, the phyla, just appear in the fossil record, fully formed, seemingly out of nowhere, without clear evolutionary ancestors preceding them. Even the early discoveries, like Walcott's Burgess shale finds back in 1909, they just highlighted the suddenness. None of the expected precursors were there.
But wasn't the explanation always that the fossil record is just incomplete? That we just haven't found the ancestors yet?
That was Darwin's argument, yes. He called the record extremely imperfect. But paleontologists today, people like Stephen Jay Gould, Niles Eldredge, they argue the record is actually pretty decent now. It's adequate, they'd say. And they suggested that the gaps we see, these sudden appearances, they might actually reflect real events in life's history, not just missing data.
So these explosions aren't just a Cambrian thing, it happens elsewhere.
Oh, yeah, it's a pattern you see again and again. Major fish groups, the first land plants, flowering plants later on, even groups like mammals and birds show these relatively rapid bursts of diversification, these explosions, rather than slow, steady gradualism.
Okay, and what about the classic examples we hear about, like whales evolving from land mammals, those
Yeah.
transitions?
Even those which are often presented as kind of the poster children for macroevolution face some, well, significant challenges when you look closely. Think about all the changes needed. A blowhole appearing and migrating to the top of the head, forelimbs becoming flippers, developing insulating blubber, changing the whole reproductive system for underwater birth. It's a lot.
Right, those aren't just small tweaks.
Not at all. Biologist Richard Sternberg looked at the math and the genetics required, and considering the relatively short geological time frame they supposedly happened in less than 10 million years, he called it too many genetic rewirings, too little time. And it's similar for human origins. You have the ape-like Australopithecus fossils and then the human-like Homo fossils. But there's what many paleontologists describe as a large, unbridged gap between them.
So not a smooth transition.
Apparently not. Ernst Mayr, a really major figure in evolutionary biology, noted quite frankly, not having any fossils that can serve as missing links, we have to fall back on the construction of a historical narrative.
So if the fossils don't always show those clear, gradual branches, what about our genes? DNA evidence. Surely that should give us a clear tree of life, right?
Well, that was certainly the hope. Which leads us right into problem 4. Molecular biology has, surprisingly often, failed to produce a single, consistent, grand tree of life. The initial expectation, especially with the rise of genomics, was that DNA would clearly map out how all organisms are related through universal common ancestry. One big, happy family tree.
But it didn't quite work out that way.
No. As the journal New Scientist bluntly put it some years back, comparing different genes often told contradictory evolutionary stories.
I mean, if you look at gene A, it suggests one family tree, but gene B suggests a different one.
Exactly. Carl Woese, a pioneering microbiologist, observed that these phylogenetic incongruities, these disagreements between gene trees, can be seen everywhere in the universal tree. It messes up the neat picture. And this isn't just a problem down with microbes where they swap genes horizontally, which complicates things. You see these conflicts even in higher organisms.
Like mammals.
Yeah, for instance, there have been studies using specific types of RNA molecules, microRNAs, that suggested a radically different diagram for mammals, one where humans might be grouped closer to elephants than to rodents.
Whoa, that's very different from the usual tree based on anatomy.
It is, and it directly contradicts those traditional truths based on morphology, on physical characteristics. This kind of conflict between molecular data and morphological data is actually surprisingly common. Some sources even describe the whole tree of life concept as lying in tatters because of this onslaught of negative evidence.
Okay, so the big takeaway from fossils and genes seems to be life's history looks less like a neat, gradually branching tree and more like, well, sudden bursts of novelty and maybe a tangled thicket of conflicting genetic relationships. It's a much messier picture.
Mm.
Right, let's get really granular now. Let's look inside the cell at the actual genetic material, DNA, and the proteins it codes for. This is where the core engine of evolution, random mutation, and natural selection is supposed to be doing its work, building complexity step by step.
Okay, and here we encounter more scientific challenges, starting with problem 5, genetics. The issue is that mutations, the supposed raw material, overwhelmingly cause harm or are just neutral. They don't seem very good at building complexity. The standard neo-Darwinian idea relies heavily on random mutations creating new, useful variations for natural selection to then favor.
But the argument is they mostly break things.
Or do nothing significant, yeah. While mutations definitely happen, the critique is that there's very limited evidence they can actually generate the brand new, complex features needed for major evolutionary jumps, what we call macroevolution. They're maybe good at tinkering with existing systems, but not inventing fundamentally new ones.
Okay, so if mutations aren't great builders, that connects to the next point, right? About complexity itself.
Exactly. It ties directly into problem 6, biochemistry. How can unguided random processes produce the staggering complexity we see inside cells? This is where the concept of irreducible complexity comes in. Biochemist Michael Behe coined that term. He used it to describe biological systems that require multiple specifically matched parts to be present at the same time to have any function at all.
Like the mousetrap analogy.
Precisely. Take away the spring, or the hammer, or the holding bar. And the trap doesn't just work poorly, it doesn't work at all. It's useless until all the core parts are there and working together. That's irreducible complexity.
And Darwin himself actually acknowledged this kind of possibility, didn't he?
He did. He famously wrote that if any complex order existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down.
And the bacterial flagellum is often used as an example.
It is. It's this tiny outboard motor that bacteria use to swim. Incredibly complex. It requires about 30 different proteins working together in a precise way. If you remove just one of those key proteins, the whole motor fails. It can't be built step by step with each step providing an advantage, the argument goes. And lab research, like work from Joe Thornton's lab on protein evolution, seems to back this up. They tried to experimentally rewind proteins to their supposed ancestral forms. Often these resurrected ancestral proteins were non-functional.
So going backwards didn't work.
Right, and it suggested that evolving new functional proteins likely requires navigating through a whole series of non-functional intermediates, needing exceedingly unlikely, extraordinarily lucky sequences of mutations to get there. It highlights the difficulty of building new protein functions from scratch via random changes.
It also seems like a lot of things that were once thought to be useless in biology turned out not to be. That connects here too, right?
Absolutely. Which brings us neatly to problem 7, inaccurate Darwinian predictions about so-called vestigial organs and also junk DNA. These were often held up as evidence for evolution as leftover bits. For decades, evolutionary biologists pointed to things like tonsils, the appendix, the coccyx or tailbone, even the thyroid gland as vestigial remnants of our evolutionary past, no longer needed.
I remember hearing about the appendix being useless.
Right. During the Scopes trial back in 1925, one biologist claimed there were over 180 such useless structures in the human body.
Over 180, wow. But we now know that's, well, wrong.
Completely wrong, basically. Science has since discovered important functions for almost all of those structures. Tonsils are key parts of the immune system. The coccyx is an anchor point for muscles. The thyroid is crucial for metabolism. And the appendix. Far from useless, it's now thought to be a reservoir for beneficial gut bacteria and plays a role in immune function. As New Scientist noted back in 2008, biologists are now extremely wary of talking about vestigial organs at all.
The whole idea kind of backfired then. And the same sort of story happened with junk DNA.
Exactly the same pattern. For years, the roughly 98% of our genome that doesn't directly code for proteins was labeled junk DNA. It was presented as evidence of evolution's messy, unguided process, just leftover evolutionary debris.
But then came the ENCODE project.
Right, the Encyclopedia of DNA Elements Project, a huge international effort. And in 2012, they published results showing that the vast majority, at least 80%, and potentially close to 100% of the human genome is biochemically active. It's doing something.
So not junk at all.
Not junk at all. As one ENCODE lead scientist put it, almost every nucleotide is associated with a function of some sort or another. Even things called pseudogenes, once thought to be broken, non-functional gene copies, are now being recognized as playing sophisticated roles in regulating gene expression. They have functions, they're conserved, meaning natural selection is likely preserving them.
It really shows how assumptions can maybe hinder scientific progress sometimes. Assuming it was junk meant people didn't look for function for a long time.
Precisely. It's a good example of how a theoretical assumption, in this case based on Darwinian thinking, potentially slowed down discovery.
Okay, so the key insight here seems to be that looking inside the cell, the core Darwinian mechanisms face big challenges in explaining the construction of complexity. And past arguments based on supposedly useless biological features have often been overturned by new discoveries revealing function. Let's zoom out again. Let's look at the global picture, how species are spread across the planet, and also how similar features keep popping up in unrelated organisms. This creates some puzzles too, right?
It certainly does. This leads us to problem 8, convergent evolution. This phenomenon challenges Darwinism in some interesting ways and potentially undermines the core logic behind using similarity to infer common ancestry. See, a basic idea in building evolutionary trees is that similarity implies relationship. If two animals share a feature, it's likely because they inherited it from a common ancestor.
Makes sense, like cousins resemble each other.
Right, but convergent evolution is where organisms that are not closely related independently evolve very similar traits.
Like wings in bats and birds and insects. They didn't all get them from one winged ancestor.
Exactly, that's a classic example. But it goes much deeper and happens far more often than you might think. Complex camera-like eyes, for instance, are thought to have evolved independently maybe 40 to 65 times.
Wow, that many.
Yeah. Bioluminescence, the ability to produce light, maybe over 50 independent times. Echolocation, like sonar, evolved separately in bats and dolphins. And what's really fascinating is that this convergence sometimes happens right down at the genetic level, finding similar genetic sequences or mutations driving similar traits in unrelated lineages. For instance, some studies found bird mitochondrial DNA unexpectedly resembled that of snakes and lizards more than other reptiles.
So similarity doesn't always mean common ancestry. It can mean common design solution. Or similar evolutionary pressures leading to the same result.
Well, that's the debate, isn't it? It shows that similar structures can arise independently, which means you can't automatically assume similarity equals shared inheritance. It forces biologists to make what are sometimes called ad hoc appeals, special explanations for why these similarities aren't due to common ancestry in these specific cases, to preserve the overall tree structure.
Okay, that's convergence. And then there's the issue of just where species live. Biogeography.
Right. Problem 9. Neo-Darwinism struggles to explain the biogeographical distribution of many species. Evolutionary theory predicts that where species are found should generally match their proposed evolutionary history and the movements of continents, for example. But as one source puts it, this expected continuity is simply not there in many cases.
Like what? What are the puzzling cases?
A really big one is the origin of South American monkeys, the Platyrians, or New World monkeys. They're thought to be descended from African monkeys, Old World monkeys.
Okay, makes sense. Monkeys look like monkeys.
But here's the kicker. They supposedly diverged around 30 million years ago. However, Africa and South America had already drifted far apart. By then, they separated maybe 100 to 120 million years ago. And South America was an isolated island continent for a very long time.
So, how did the monkeys get from Africa to South America across a massive ocean, long after the continents split?
Well, that's where the rafting hypothesis comes in. The idea is that monkeys, maybe clinging to large mats of vegetation washed out to sea, somehow survived a journey of potentially thousands of kilometers across the open Atlantic Ocean.
Rafting monkeys. Seriously. Across the Atlantic.
Seriously, experts themselves admit the scenario is truly amazing for a mammalian order. Highly improbable, implausible, even suspect. Yet, because common descent from African monkeys is assumed, rafting is often presented as the most likely explanation, however unlikely it sounds.
That sounds like a stretch.
It does, and similar, highly improbable oceanic dispersal stories are invoked to explain other odd distributions, like certain lizards and large rodents arriving in South America, lemurs getting to Madagascar, elephant fossils found on remote islands, even freshwater frogs somehow hopping between isolated oceanic islands. These biogeographical puzzles strain the standard explanations.
Okay, so the big picture here is that convergence, similar designs popping up independently, complicates the simple similarity equals ancestry rule. And the geographical distribution of species sometimes requires invoking these incredibly unlikely, almost fantastical cross-ocean journeys to fit the assumed evolutionary narrative. We've covered a huge amount of ground today, from the origin of life chemistry through fossils, genes, cellular complexity, vestigial organs, convergence, biogeography, quite a list of challenges. So what does this all mean for the bigger picture? Are these just minor issues being worked out, or is something more fundamental stirring in evolutionary biology?
Well, connecting all these dots, our sources suggest there's definitely something stirring. Some are calling it an approaching storm in evolutionary theory. You find quite a few scientists, including many who absolutely believe in evolution in a broad sense, expressing frustration about the lack of debate concerning the problems with the standard neo-Darwinian model. They feel these issues aren't getting the attention they deserve.
So there's a feeling that maybe the standard story is presented as more settled than it actually is.
Exactly. Major publications, even the Guardian newspaper in 2022, ran articles asking things like, do we need a new theory of evolution? They noted that textbooks often give an absurdly crude and misleading picture of how it all works.
A new theory, that's a pretty big statement.
It is. Some scholars are arguing that neo-Darwinism must mutate to survive. Researchers like Olin Brown and David Hollander point to the sheer statistical improbability. They calculated odds like 10 to the power of negative 50 for the kind of large-scale genetic changes needed to build genuinely new, complex structures. You even have philosophers of biology like Jerry Fodor and Massimo Piatelli-Palmarini, who wrote a whole book called What Darwin Got Wrong in 2011, stating pretty frankly, we don't know what the mechanism of evolution is.
Wow. And even Darwin himself is being looked at differently now.
Yeah, that's another interesting thread. Some scholars are reevaluating Darwin's original work and arguing that maybe he wasn't quite the evolutionist in the modern sense that we often assume. They suggest the modern theory, particularly the strong emphasis on natural selection as the sole or primary engine, has kind of projected its own ideas back onto Darwin. They point to his sometimes convoluted style of argumentation and suggest he still held onto a more static worldview than we credit him with. The idea of natural selection being the absolute core of his theory might even be a modern invention, they argue.
So there's internal debate about the mechanism, the scope, and even the history of the theory itself.
Absolutely. This persistent discontent, as some call it, likely stems from just how incredibly complex biology is. New data is always emerging, and it can easily challenge existing frameworks. There's this inherent tension, you see, between trying to describe evolution as this dynamic, often unpredictable process and the scientific need for clear, testable models. And it's worth mentioning, there's a formal statement signed now by over 1,200 PhD scientists from various fields expressing skepticism about the creative power of random mutation and natural selection alone to account for life's complexity.
And these are mainstream scientists.
Yes, PhD scientists.
Oh.
They're urging a more careful examination of the evidence for Darwinian theory. It's important to see this not as an attack on science itself, but as a call from within science for more rigorous critical thinking and openness to exploring alternative explanations or modifications to the theory.
Wow. Okay, that was a truly expansive deep dive. We've gone through so many different layers of the ongoing scientific conversation around evolutionary theory. It really becomes clear that the story of life's origins, its development, it's definitely not a closed book. Scientists are still grappling with some really profound questions and constantly refining, sometimes challenging our understanding.
Indeed. And the discussions we've touched on today, from the chemical hurdles at the start to the fossil record surprises, the genetic tangles, the amazing complexity inside cells, the biogeographical riddles, they all just underscore that science isn't some static collection of facts. It's a dynamic process, isn't it? It's about inquiry, discovery, debate, and constant revision. The very fact that these robust debates are happening among experts, that there are calls for potentially new frameworks for evolution, that really shows the scientific method in action. It's about questioning, testing, and adapting based on evidence.
Absolutely. That leaves us, and you, our listener, with a pretty big question to ponder, doesn't it? Given these persistent scientific challenges, these ongoing debates within evolutionary biology that we've explored today, How do you think our understanding of life's history might change in the coming years or decades? What kinds of new discoveries out there could fundamentally reshape what we believe about where we came from? And what might new scientific models, perhaps going beyond the standard framework, look like as we try to better explain the incredible, intricate complexity of life?